This is Unit 2.2, where we're going to be looking at the biomolecules. So the four types of biomolecules are carbohydrates, proteins, nucleic acids, and lipids. And you should remember from Unit 1 that the six most common elements that are found in these four biomolecules are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. So the term biomolecule means that these are large molecules that are built up of smaller subunits. In other words, they're polymer, poly meaning many, made up of repeating subunits called monomer. Mono means one. So you need to know not only the four biomolecules, but the monomers for each of those. So proteins are polymers that are made up of amino acids. Nucleic acids are made up of nucleotides. Carbohydrates are made of saccharides, and lipids are repeating subunits of carbon chains. They have a bunch of different forms, and we're going to go over them a little bit, but they're basically carbon chains that are put together. Which of the following molecules makes up or composes proteins? These are joined together to form carbohydrates. So carbohydrates are made up of those saccharides or sugars, and a sugar contains carbon, hydrogen, and oxygen in a very specific ratio, 1 carbon to 2 hydrogens to 1 oxygen. So the main purpose of carbohydrates is to provide energy, glucose being the molecule that drives cellular respiration, making ATP. You can also have complex carbohydrates that provide support. So chitin is found in shells and shells of insects, and also in mushrooms. Cellulose is found in plants. Their cellulose especially is not digestible to humans or to any mammalian enzymes. So this is just to show you these rings that are strung together. So polysaccharides are the next biomolecule is lipids. Lipids include fats, which we're familiar with, that we eat, phospholipids, which are found in the cell membranes, steroids, and waxes. So without getting into too much detail about their structures, they can come in a lot of different forms, but the main purpose of fats is to store energy. The reason for this is that fats contain 9 calories per gram, as opposed to all the other biomolecules are less than half of that. They all contain 4 calories per gram. So by storing energy more densely, and that energy is stored in the carbon-carbon bonds, it allows organisms to store more energy with less mass that they have to move around. So you'll see energy stored in the form of fats for organisms that have to move around. Those organisms that don't move around tend to store energy as they're made in carbohydrates in the form that they're made in. So the other really important thing that lipids do is they compose the cell membrane. So those phospholipids, and I'll show you a picture in a second, have 2 parts that allow them to form a bilayer that controls what can move in and out of the cell. So before I show you what a phospholipid looks like, I just want to make sure that you know that hydrophobic means a fear of water, or it doesn't get along with water. And hydrophilic is a love of water, and it will get along with water or any aqueous substance. So phospholipids contain a glycerol, 2 fatty acids, and a phosphate group. And you can see you have your hydrophilic head and your hydrophobic feet. And they line up because it's a bilayer. There's 2 layers where their heads are in the extrastitial fluid, and The head from the other layer is in the cytoplasm. So this allows the membrane to prevent water from coming in an uncontrolled way, and it prevents materials from going in and out. So you can see in this picture of the cell membrane, you have channels, you also have structural proteins, and it allows material to be controlled going in and out of the cell. Waxes are solid at room temperature and hydrophobic. These are found on the leaves of plants, on the top surface of leaves, and plants put these waxes together to help prevent water from evaporating from them. And then our fats, which we talked about, you can have saturated versus unsaturated fats. Unsaturated fats have less hydrogens because they have a double bond. And you see the lower one is an unsaturated. The saturated means that it's saturated with hydrogens. So those hydrogens are on every carbon molecule. Unsaturated means it's not saturated with hydrogens. So that double bond prevents those hydrogens from going onto each carbon. So phospholipids are the main component of hydrophobic means something. Hydrophilic means something. A surfactant means that something is breaking up the hydrogen bonds of the water, and it has it's amphoteric, so it has a part that is hydrophilic and a part that's hydrophobic. So it allows materials that are normally hydrophobic to be basically dissolved in the water, so they can be carried away. An example of a surfactant would be dishwashing detergent that can take grease out of a pan. Then you have your steroids. So both estrogen and testosterone are examples of steroids. Your norepinephrine is another steroid. Cholesterol is a steroid fat that allows membranes to be fluid. Why would it be important to know if you have high cholesterol? All of these are examples of lipids except when fatty acids cannot pack together tightly enough to make a solid fat, they have lipids are the next biomolecule are proteins. So these are the rock stars of the biomolecule world. They do everything in cells. Enzymes are proteins, and they have an unlimited number of structures. So they form three-dimensional shapes, and I'll talk about that in a second. But these three-dimensional shapes allow them to perform functions, such as breaking things apart or putting things back together. They're used for transport, carrying out chemical reactions, communication. They're very diverse, and there's your 20 amino acids, and they're kind of like the letters of our alphabet. So there's no limit to the number of words and sentences that you can put together that are novel and unique and have different meanings. And that's true with proteins. There's no limit to the number of amino acids that you can put together that then form these unique proteins. And the way that they're put together causes the different configurations in 3 dimensions. So it allows these different 3-dimensional structures that can perform tasks. So the takeaway, a protein is a molecule, remember, made up of amino acids. That's the monomer. Amino acids, when they're formed together, when they're linked together, are linked by a peptide bond. These peptide bonds are formed by dehydration synthesis. So you have your hydrogen in the amine group, and you have your hydroxide on the carboxyl group, and they both come off and they form that peptide bond. So you're left with a water, which is how it gets its name, for dehydration synthesis. So you can watch this video. For each type of protein, there are different levels of structure. The primary structure is just a sequence of amino acids. So there's no three-dimensional shape. It's just those R side chain, which gives each amino acid its unique identity, are in a different sequence. So then when you have those R side groups interacting with each other, you can have coils and folds. So all of these interactions for the secondary, tertiary, and quaternary structure are all caused by hydrogen bonds. So remember, hydrogen bonds aren't true bonds. They're intermolecular forces that are found when you have polar molecules. So it kind of acts like magnets. So they can be disrupted rather easily. And they're more like Velcro than a true bond because they can come apart and go back together again. So the secondary structure are your coils and folds. Your tertiary structure is the overall shape of a single chain of amino acids. Now, the quaternary shape is when you have multiple single chains of amino acids that come together and form an even bigger structure. So here you can see your primary structure is just a sequence of amino acids. Your secondary structure are your alpha helices and your beta pleated sheets. Your tertiary structure is a three-dimensional structure of one single protein, and your quaternary structure are multiple proteins that are associated with each other. And don't forget that R group gives each amino acid its unique properties. There's some pictures in your book. The carboxyl end of one amino acid binds to the amine group of the next amino acid. This synthesis removes a water molecule. What is it called? So here you just see you have your hydroxide from the carboxyl group and your hydrogen from the amine side, and they'll come apart and you get a water and a peptide bond is formed, and that is called dehydration synthesis, which gives each amino acid its unique properties. The amino group of one amino acid is linked to the carboxyl group of another amino acid. This link is A. The overall shape of a single chain of amino acids in a protein is that protein's primary, secondary, tertiary, or quaternary structure. The overall sequence of a protein's amino acids is its. What type of reaction is used to join monomers together by removing a hydrogen ion and a hydroxide ion? The next biomolecule is nucleic acids. These are made up of nucleotides, and a nucleotide has three parts: the sugar, the phosphate group, and the nitrogenous base. So in DNA, the sugar is deoxyribose. In RNA, it's ribose. The sugar is ribose. So both of those nucleic acids are named for their sugars. And then you have ATP, or adenosine triphosphate, which is a nucleotide, not a nucleic acid because it's a single unit. It has the same structure, though. It has three phosphates. So the tri means three. The high energy state of this is ATP, where you have three phosphates. So what happens in cellular respiration, in order to force that phosphate onto the phosphate group of ADP, adenosine diphosphate, glucose is broken down, and the energy in the carbon bonds is used to force that phosphate onto that. It's a high energy bond. It's relatively unstable, but it can be used to drive functions in the cell. ATP stores energy in the bonds between its. So just to summarize, proteins have 4 calories per gram. The repeating subunit is amino acids. They do pretty much everything in the cell. They are enzymes. They form structures. And in the body, as opposed to importance in the cell, They're used for structures, they're enzymes, and you can find them in foods such as meats, beans, and nuts. And you can see that structure right there. So you can see also that R group, that top picture shows just the R chain, but alanine has a methyl group. Valine has 3 of those, and you can see how they differ, and that side chain is what gives them their identity and their properties. Carbohydrates have 4 calories per gram. They're made up of monosaccharides. You can see the structure there. And in the cell, they are cell surface markers. They're used for energy and also labeling materials for exports. They make up the name tags for things. Why are they important in the body? They're an energy source. They also help recognition of non-self. So this is how your body can see that you are being invaded by a parasite, a virus, a bacterium. And they're found in breads, cereals, grains, and tubers. Nucleic acids are the next biomolecule. They have 4 calories per gram. They're made up of nucleotides. Remember, they have the sugar, the phosphate group, and the nitrogenous base. They really only have one function: to carry genetic information, to carry information. That genetic information is actually the recipes to make proteins. And then ATP obviously is the energy currency of cells. So why are they important in the body? All cells obviously have DNA and ATP, and they're found in everything we eat. So you don't see that on, your list of ingredients for the foods that you eat, your nutrition labels. But as long as you're eating something that's organic, it has cells, and therefore it has nucleic acids in it. And our final biomolecule is lipids. This has 9 calories per gram. It's made out of chains of carbon and hydrogen. And it's important because it makes up the cell membrane, and also long-term energy storage. So they're important in the body because they're used for cell membranes, again, long-term energy storage and insulation. They're found in butter, nuts, fish, some legumes. So remember, cell respiration is not breathing. It's using glucose, which is a carbohydrate, to create ATP, which is a nucleotide. And ATP is then used to do everything in the cell. So remember, the waste product of this is carbon dioxide and water. So energy is stored in the high energy phosphate bonds of ATP. And ATP is recycled. So when the phosphate gets broken off and energy is released, you have ADP and a phosphate. And then cell respiration breaks a glucose molecule to form that phosphate back on that ADP, which turns it into ATP. The cell membrane is also important because it controls influx and outflux. So making sure that the cell doesn't lose things or have things come into it that it doesn't want, the cell membrane allows control of that. So it's composed of a phospholipid bilayer, when we already talked about those phospholipids. So the hydrophobic tails are in the middle, and the hydrophilic heads are on the outside, where they're going to find those aqueous solutions. And then you have materials embedded into the cell membrane, such as cell surface markers, which act like a name tag. They're usually glycoproteins, which are carbohydrates attached to a protein. You can also have receptor proteins, which allow materials on the outside to change things on the inside. So you can see this is an example of a neuron, where you have a neurotransmitter in the synapse, and you have your receptor protein. When that neurotransmitter lands on that protein, it changes the configuration on the inside without that neurotransmitter actually ever entering into the cell. Then you can have transport proteins. So because substances can't pass directly through the bilayer, but they may need to get in, you have these transport proteins. So you can have a pore, which is kind of like just a tunnel that allows things to go through. You can have movement of substance through it pretty quickly, because there's nothing controlling it other than traffic going through. Then you can have a carrier protein, which kind of acts like a gateway. If you're going into a turnstile to get into an amusement park or something like that, where you can only have one molecule at a time entering in the cell. So this can control the amount that enters or exits the cell. And you could have enzymes that are embedded into the membrane. Remember, enzymes are catalysts that cause a reaction to occur at a lower energy. And your catabolic is breaking something down, your anabolic is putting something together. And there's really two types of transport: passive, which does not require ATP, and active, which does require energy. So some examples of passive transport is anytime you have something moving down its concentration gradient. In other words, going from an area of high concentration to an area of low concentration. If you sprayed air freshener in a room, what would happen? It would be very concentrated and then it diffuses throughout. That's an example of something going down its concentration gradient, where it goes from an area of high concentration and attempts to make the concentration the same everywhere in the solution or area. So the passive transport that we need to know is simple diffusion, where you have material that can pass directly through the lipid bilayer of the cell membrane. Oxygen can do this, carbon dioxide also. Steroid hormones can go in and out of the cell without having to go through a pore or carrier protein. Facilitated diffusion is when you have material that can't pass directly through the cell membrane. So you'll have transport proteins that allow this material to still go down its concentration gradient, go from an area of high concentration to low concentration, but it can't pass directly through the cell membrane. So you can have the channel proteins that we talked about. Those are like tunnels. And then your carrier proteins, where it allows one at a time. Again, all of this is still passive transport. Osmosis is also another example of passive transport. You need to be certain of the definition of osmosis. It's water that passes through a selectively permeable membrane to have the concentration be homogeneous. So it's going down the concentration gradient from an area of high concentration to low concentration, but it has to go through a cell membrane, and it's only movement of water, not solute. So just kind of comparing the difference between diffusion and osmosis, both of them equalize the concentration of two solutions from an area of high concentration to an area of low concentration. Diffusion, you can have any solute. It can be gas, it can be liquid. No membrane is involved. Osmosis is only movement of water, and it has to be through a semi-permeable membrane. So the different types of osmotic solutions that you can have a hypertonic solution, where you have more solute outside of the cell, and water is going to attempt to leave the cell to try to make the concentration of water equal on both sides of the cell membrane. So in a hypertonic solution, you will have net movement of water out of the cell. If it's isotonic, you have equal concentrations both outside and inside the cell, and you have a net water movement of 0 because the same amount of water is moving in as is moving out. And in a hypotonic solution, you have a lower solute concentration in the cytoplasm. So you have higher inside the cell. So water is going to go into the cell to try to diffuse that lower concentration of water. So the cell will swell and burst. So in plants, rigid cell walls of the plant cells kind of prevent a cell from really deforming too much and bursting. That doesn't mean that it can't. It's just not as likely as in an animal cell. So those were all passive transport. And then we have active transport. This requires ATP because you're moving substance against their concentration gradient. So you are going from an area of high concentration to an area of high concentration, or low concentration to low concentration. So you're not allowing the material to form a homogeneous concentration solution. So you can have a pump. The most famous one is a sodium-potassium pump, which allows all neurons to fire. You don't have to know this too much, but you're moving sodium and potassium against their concentration gradients. And then when they go back to they go down their concentration gradients, that's what powers the reactions. You can also have vesicles. We learned about those when we talked about the structure of the cell, which is a small membrane-bound sac. So if a cell is taking something into it, it's called endocytosis. And you can see right here how you have this little Pac-Man, and it takes the material and encloses off, and now that material is inside the cell. Exocytosis is when a cell needs to export something, and you have a vesicle from the Golgi apparatus that fuses with the cell membrane. And this is the fluid mosaic model, how this works, where you have those phospholipids that kind of glide over one another. And then the cell can export that material. You can also have signals that we talked about, and you can have either long distance or neighboring cells. Which of the following is not a large biomolecule? Which of the following is a carbohydrate? The two types of nucleic acids are? Which of the following molecules are linked together to form nucleic acids? What type of molecule is ATP? DNA stores the two strands of a DNA molecule are held together by a covalent bond. Means what type of carbon-based molecule is the first to be broken down by the body's cells to provide a source of energy? Think cell respiration. Carbon dioxide is converted into organic compounds in the Unlike other carbon-based molecules, nucleic acids are known for their What is this? What is this?